Let us discuss about a female reproductive organ, that is gynecium. That gynecium is the innermost whorl of the flower. It is made up of individual units. Those individual units are named as pistil or carpel. Gynecium do have individual units named pistil or carpel. And pistil or carpel, they are varied in number from species to species. If there may be a single carpel, then it is said to be monocarpellary. Monocarpellary. If there are two carpels, bicarpellary. Tricarpellary. Even multicarpellary. Based on the number of carpels. Monocarpellary, bicarpellary, tricarpellary and multicarpellary. It means if the single carpel is there, that is monocarpellary flower. If there are two, bicarpellary. If there are three, tricarpellary, and more than three, that would be multicarpellary. Based on the number monocarpellary, bicarpellary, tricarpellary and multicarpellary. If it is monocarpellary, it is present free only. But if it is bicarpellary, tricarpellary or multicarpellary, there will be two conditions. If those carpels, if they are two or more than two, if they are free, then it is said to be apocarpous. When two or more than two carpels are there and those carpels are free, the condition is said to be apocarpous. And carpels which are two or more than two, but they are in fused condition, it is said to be syncarpous. Syncarpous. It is said to be syncarpous. And now, if you analyze the structure of pistil or carpel, we do recognize three parts. First one, stigma and elongated style and oval ori. These are the three distinct parts of carpel or pistil. This stigma is a flat form to receive the pollen grains. That stigma is a flat form which receives the pollen grains. And the style, very long and slender structure with the lumen inside. When the pollen grains fall on the stigma, the pollen tube will form. That pollen tube pierces through the style and goes to ori. And the ori is a structure having orion tissue. In that orion tissue, there will be a megasporangium or ovule. There may be a single megasporangium or ovule as in case of cereals. Even there are innumerable number of megasporangia and ovules in orion tissue. They are there in ori as well. And that megasporangium in which there is a development of female gametophyte where the female gamete is there. Many such Megasporangium or ovules are there in ovary. In the ovary, there are one to many Megasporangia or ovules are present. And let us see the structure of ovule or Megasporangium. 80% of plants do have a bent ovule, and such ovule is named as anatropous ovule. Anatropous ovule. In 80% of plants, the ovule is bent, that is named as anatropous ovule, and in addition to anatropous ovule, we do have totally 4 types, but in 80% of them, anatropous ovule is there, and remaining 3 are orthotropous ovule. Orthotropous ovule means the ovule is straight, and an ambitropous ovule, where ovule body and funicle, they are at right angles to each other. And a campylotropous ovule is one where it looks like horseshoe shaped. And an anatropous ovule is a bent ovule. We have to study the structure of this anatropous ovule only. If they ask the structure of Megasporangium or ovule, you have got to draw this diagram and explain. In ovarian tissue, there is a placenta. This ovule is attached to the placenta by the stalk. That stalk is named as funicle. That stalk is named as funicle. With the help of funicle, this ovule is attached to the placenta in ovarian tissue. Here, the ovule body is attached to the funicle. The place where ovule body is attached to the funicle, that junction is said to be hilus. That is the part where ovule body is attached to the funicle. And now, the ovule body at its internal position having homogeneous tissue named nucellus. And this total nucellus is covered by 2 coverings. Those coverings are named as integuments. Those 2 coverings are named as integuments. Those are, this is outer integument and this is inner integument. These are the 2 coverings which cover around the central homogeneous tissue nucellus. At one side, free end, these integuments are discontinuous. That discontinuous area is named as micropyle. That discontinuous area is named as micropyle. And opposite to the micropyle, this end is named as chalaza. It means in ovule we do have 2 ends, micropylar end and a chalazal end. Micropylar end where micropyle is there, both the integuments are interrupted, discontinuous. That part is named as micropyle. And opposite to this micropylar end is chalaza. In nucellus, towards micropylar end, there is a sac-like structure. This sac-like structure is named as embryo sac. Embryo sac. That sac-like structure is named as embryo sac, and this embryo sac is a female gametophyte where egg or female gamete is there. Just like pollen grain is a male gametophyte, in the similar way, embryo sac is female gametophyte. And this embryo sac at mature is having 7-celled and 8 nucleated. It is a 7-celled and 8 nucleated. Towards micropylar end, a group of 3 cells is there that is said to be egg apparatus. Egg apparatus. And in this egg apparatus, this is egg or egg cell, and one is egg cell, and 2 synergids. 2 synergids. Towards micropylar end, a group of 3 cells are there. One is egg cell and 2 are synergids, and these 2 synergids towards micropylar end are having different thickenings. That the thickening is named as filiform apparatus. Filiform apparatus. These are 3 cells towards micropylar end, and a group of 3 more cells towards chalaza. And these 3 cells together named as antipode. Antipode. And there is a single large cell. It is having 2 polar nuclei. They are haploid in nature. In embryo sac, it is a female gametophyte. It is 7-celled and 8 nucleated structure. Those 7 cells are 3 cells in egg apparatus, 3 antipodals, 6, and a larger cell in the middle, in which 2 polar nuclei are there. 8 nucleus means 3 cells with 1 nuclei each, and these are also 3 cells with 1 nuclei, totally 6, and these 2, totally 8 nucleated. It is the structure of Megasporangium Oil, and it is anatropous ovule, bent ovule. Its stalk is named as funicle. With the help of funicle is attached to the placenta of ovarian tissue, and ovule body and funicle are joined at the junction named hilus, and the ovule body is having a homogeneous tissue inside named nucellus. This nucellus is covered by 2 coverings named integuments. Those are outer integument and inner integument. But these 2 integuments are interrupted towards free end, that is said to be micropyle, and opposite to this micropyle end is said to be chalaza. In nucellus towards micropyle, a group of 3 cells are named egg apparatus. That egg apparatus consists of 1 egg cell and 2 synergids. In these 2 synergids, a different thickening towards that micropyle named filiform apparatus, and towards the chalaza, in this embryo sac, a group of 3 cells named antipodals and a large cell with 2 polar nuclei. And this embryo sac is a female gametophyte which is 7 celled and 8 nucleated and is anatropous oil. I have mentioned here arthotropous oil where funicle, hilus and a micropyle straight. And in case of ambitropous, this funicle and oil body, they are at right angles to each other. And carpylotropous oil, where the oil is horseshoe shaped. And finally anatropous is an anatropous oil as well. We have seen the structure of oil or megasporangium. Here the female gametophyte is embryo sac, and it is developed from one of the nucellus cell which is present towards micropylar end. A cell which is present towards micropylar end in the nucellus cell is giving rise to the female gametophyte or embryo sac. Now let us see how this embryo sac or female gametophyte is developed. Development of embryo sac or female gametophyte. Development of embryo sac which is a female gametophyte. I have just mentioned totally the internally it is filled with nucellus. One of the nucellus cell gives rise to this embryo sac. Which nucellus cell? That nucellus cell which is present towards micropyle, I am going to draw here. This is a nucellus cell which is present towards micropyle, and this nucellus cell becomes megaspore mother cell. Megaspore mother cell. One other nucellus cell towards micropyle becomes megaspore mother cell. It is diploid in nature. This megaspore mother cell undergoes meiotic division, meiosis, so that from this megaspore mother cell a tetrad of 4 megaspores is formed due to meiosis. A tetrad of 4 megaspores. A tetrad of 4 megaspores is formed. All of them are haploid because meiosis took place. That's why they are all haploid megaspores. They are in tetrad, they are in association. They are formed from megaspore mother cell. And later on, among these 4 megaspores, 3 get degenerated. 1 is remained, 3 get degenerated. This is towards the micropyle. This is a single megaspore is functional and 3 are degenerated. This single megaspore now becomes embryo sac. From this single megaspore, this total embryo sac is formed. Let us go with this megaspore. It is having haploid nucleus. This is megaspore. And the nucleus, haploid nucleus, which is present inside, now divides into 2. One goes to, they go to the other ends by mitosis. And these 2 nuclei further divide to form, that is once again by mitosis, so that 2 haploid nuclei here and 2 haploid nuclei here are formed. This is 2 nucleated stage, 4 nucleated stage here. One more time there is a mitosis, so that 4 nuclei here, all of them are haploid, 4 here. And remember, as we are dealing with bent ovule or anatropous ovule, this is micropylar end, this is chalazal end. 8 nucleated stage now. During this, among these 4 nuclei, around 3 nuclei, cell wall is formed, and 1 nucleus, haploid nucleus, is remain from this. And in the similar way, among these 4 nuclei towards micropyle, around 3 nuclei, cell wall is formed, so that 1 free nucleus is remained. The structure becomes like this. This is chalaza and is micropyle. Towards micropyle there are 4 nuclei. Around 3 nuclei, cell wall is formed, leading to the formation of egg apparatus. Egg apparatus. And that egg apparatus consists of 2 synergids and 1 egg cell. Among these 4 nuclei, around 3 nuclei, cell wall is formed, and 1 free nuclei is made. And here there are 4 haploid nuclei, around 3 nuclei, cell wall is formed, and they become 3 antipodals, and one nucleus remains, will be free nucleus, and these two are polar nuclei. Embryo sac with 8 nucleated condition and 7 cell is developed. That is here. This is here. How this is formed? This is formed from one of the nucellus cell. That nucellus cell is present towards micropyle. That nucellus cell becomes, it is just enlarged and becomes megaspore mother cell. Megaspore mother cell towards micropyle. Totally, previously, internally it is packed with the nucellus only. One of the cells towards micropyle is a nucellus cell that gets enlarged and becomes megaspore mother cell, which undergoes meiosis. A tetrad of 4 megaspores are formed. Among these 4 megaspores, they are all haploid. 3 get degenerated. 1 towards micropylar end is functional, and this megaspore undergoes mitosis 3 times. Only free nuclear division. By first mitosis, we do have 2 nuclei. When mitosis takes place second time, 4 nuclei. Third time, 4 at chalazal end and 4 at micropylar end. And it is free nuclear division. Towards micropylar end, among the 4 haploid nuclei, around 3 nuclei, cell wall is formed, leading to the formation of egg apparatus. That egg apparatus consists of egg cell and 2 synergids. And 4 nuclei which are present towards chalazal end, around 3 cell wall is formed, leading to the formation of 3 antipodals. From both the sides, 1 nuclei are remained. They come to the center and become 2 polar nuclei. 2 polar nuclei. They are haploid. So that totally 8 nuclei are there, and 3 plus 3 and this 7 cells. And this is embryo sac or female gametophyte. They may ask with neat label diagram, describe the structure of Megasporangium ovule. You have to draw and explain all the parts. And another question they can ask, describe the development of female gametophyte or embryo sac. You have to draw this one and explain. And here, from a megaspore mother cell, 4 megaspores are formed by meiosis. That is said to be megasporogenesis. Megasporogenesis. Formation of haploid megaspores from diploid megaspore mother cell by meiosis is named as megasporogenesis. Just like formation of microspores from microspore mother cells in Microsporangium in anther is named as microsporogenesis. That is microsporogenesis. And formation of haploid megaspores from diploid megaspore mother cell by meiosis is named as megasporogenesis. Formation of haploid microspores from microspore mother cells, diploid microspore mother cells by meiosis is named as microsporangiasis. And this is about the structures and explanation concerned to a female gametophyte.